So first thing first on the list, we got genetics, which is DNA versus RNA. So I'm going to give my little pictures of DNA and my picture of RNA. And first off, we can see that DNA has 2 strands, which means that it's a double helix. Going over to RNA, we have 1 strand, making it a single-stranded. Then we have DNA, which is actually genetic material, which means that we got 46 chromosomes, 23 from mom and 23 from dad. And for RNA, RNA codes for proteins. So for all the proteins that we need in our bodies to function, they come from RNA. And DNA finally has base pairs of A and T and G and C, while RNA has base pairs of A and U and G and C. So we're going to move on to number 2, and we're going to move on to cells, and we're going to be talking about competitive inhibitors and non-competitive inhibitors. So in a little bit, I am going to be bringing up my little inhibitor, and there it is. Watch, there it is. Okay, so we're going to start off with our competitive, and the little green blob is going to be our substrate. And then our little dent is the active site. And that little orangey yellow ball, that is our inhibitor. And what the competitive inhibitor does is it binds to the active site. So it blocks the enzyme from getting to the active site and functioning. For our little non-competitive, over here, nicely drawn. Oh wait, no. Oh, I spelled it wrong. Oh, okay, there we go. Clean that up. Yes, non-competitive. So for our non-competitive, we again have the substrate, which is this nice green blob. And our active site is that little white dent. And again, our little yellow-orange ball is going to be our inhibitor. So what the non-competitive inhibitor does is that it binds to another part of the substrate, which then changes the active site. So enzymes with the receptors meant for the original active site cannot bind to it. That means the enzymes can no longer function. So moving on, we're going to go to number 3, which is evolution. And we're going to be doing genetic drift and gene flow. And we're going to start with genetic drift. And we have a population of pinks and a population of orange. And genetic drift is when the allele frequency is reduced by a cause. So let's say my finger, is an earthquake, and boom, earthquake destroys. Now the population has been reduced and the allele frequency has gone down. So we're going to move over to gene flow. And the deal with gene flow is that we're going to have 2 populations, 1 pink and 1 orange. And gene flow is actually just when individuals of a population move through immigration or emigration. So the moving over of pinks is the immigration, or with an E, and then immigration is going to be the orange, which now have accepted the pinks. So that is immigration with an I. So we're gonna lastly move on to our last unit of ecology of immigration versus emigration. And we're gonna start with immigration, and as you can see, I have drawn 2 populations, 1 blue and 1 green. Immigration with an I is the joining of new individuals into a new population. So what is going to happen is that our little blue species, or whatever, our population, they are going to go into the green ones, and now they joined the new green population. Then it's going to be a fiesta because now both colors are joined together. For emigration, again, we're going to have our 2 population of our blues and our greens. Emigration is the actual movement. So the movement of the blues into the greens is what is emigration with an E. So we're going to have our blues in a little bit come over and join the greens. So look, now they're going to move into the green. That is what emigration is.